These are the Diagnostics lightning talks, and I'm going to introduce them. So these presentations are going to review some of the newest developments around Lyme disease diagnostics. And there will be Q&A at the discretion of the speakers, time permitting. And if there isn't time, all of the speakers, of course, have agreed to talk with you after the session. And there will be microphones stationed around the room for questions. And I kindly ask you to keep your questions brief because there's only going to be a few minutes for each talk. So our first speaker, I'm delighted to introduce Stephanie Gervaisi. She's Senior Research Scientist and Data Science Consultant at the Lyme Disease Biobank, and she will present Applying Predictive Analytics to Aid Lyme Disease Diagnosis. Please help me in welcoming Dr. Gervaisi.
I think we're okay. All set. Okay. So I'm really grateful to be here with you today. This is the first time I'm attending this conference, so I'm learning a lot. It's very exciting. And I should say that I'm co-presenting this talk about predictive analytics with Dr. Liz Horn, who is the principal investigator of the Lyme Disease Biobank. So I want to say a few words about the Biobank. It's an incredible resource. There are right now over 800 participants that are currently voluntarily enrolled in this study. And increasing every day with the addition of new sites. And these participants include individuals with early acute Lyme disease with and without erythema migrans rash and annular expanding rash, those with suspected Lyme disease, people who may have seen a tick or suspect that they've been bitten, and endemic controls, which is very rare. So there are also people who are healthy people from the same regions, the same sites where the samples are being collected. And the samples are well characterized. This is also very, very unique. So all of the samples, including the endemic controls, are undergoing very rigorous laboratory testing, including what's considered the gold standard. It's serology and qPCR. And we know that there are other ways. We'll talk about that in just a moment. And so in addition to the samples that are provided to the biobank, the blood, the tissue, and urine samples that are provided, which are incredibly valuable for use by other scientists who are developing diagnostic techniques, there's also really rigorous survey data collected about these individuals. And so I've highlighted some of the features or some of the attributes or traits that are involved in the surveys. So really extensive list of symptoms. So instead of this being an open question, it's very specific. Have you experienced these symptoms, which is very useful. Medical history, including both previous tick-borne and other chronic and infectious diseases that people have experienced. Detailed encounter information, geographically and on the body. Medication use, so both over-the-counter and prescription medications. Demographics, and then really extensive notation about the rash, including photographs of the rash if they're present. So this data is actually what is the food for or the fuel for these predictive models or the predictive analytics that we're using. They are the traits, they are the input that's going in, and this predictive modeling involves training a mathematical algorithm or group of algorithms to classify input data or assign a label. So we're using classification. And so the more information we can collect about these these participants that are in the study, the better we can tune this algorithm actually to find a way to classify or label the data. And so this approach is really different than what we typically hear in frequentist statistics. So we're not looking for p-values. We're not really interested in significance. What we want to know is how well the algorithm that we train and the model that we build actually does on real-world data. So if we were to see a person that we had never seen before collect those traits, how well could we classify them, having known nothing about them before, just having been trained? And so we use a little bit of a different procedure where we subset our data into different portions, and we train, we validate, and then we test the performance of the model. So you are all familiar with this, maybe whether you know it or not. So one of the really classic classification problems is when lots of mail comes to your email box, marking it as spam or something that you actually want to read. That's a classification algorithm behind it that's working for you, thank goodness. For those of you who like to travel, if you might have your favorite site to forecast the price of flights. This is also an algorithm working behind the scenes. Of course, we all know Netflix, a little bit different, but that's a recommender system saying if you like these things, you might also like these things. But we see these approaches, these predictive analytics, increasingly used in healthcare. So I've given an example here of imaging. So there's some deep learning and neural network techniques that have been used really to see how well basically computers can find tumors compared to doctors, right? So it's really a companion diagnostic bringing the two together. So here's an example of one of those algorithms. I love trees. I love tree-based algorithms. This is an example of a random forest. It's a series of decision trees where you're basically sorting the data and partitioning it down till you get to the classification that you're interested in. I am only moving backwards. Is it possible to advance the slides back there? And one more. Thank you. So, the question I ask here and that we're asking at the Lyme Disease Biobank is whether a machine learning algorithm can help us predict the likelihood or risk of being lab diagnosed as Lyme positive. And one more. And so here I'm going to give you a couple of descriptives just to illustrate the value of these algorithms. So here we have people along the x-axis that are either Lyme negative according to the lab diagnostics or Lyme positive according to those diagnostics. And then the count or the frequency of those people along the y-axis. And so what you can see is that people who eventually test positive and who test negative in the lab present with a rash. Although we could argue that there's a greater proportion of people with a rash who eventually test positive. But if we go to this simple decision tree structure, you can see this is maybe the first level. We can sort people according to whether or not we have the rash, but that's not going to do it. It's not enough, right? We need more than that. So next slide, please. So now let's look at a subset of these individuals, so just the people who have the rash. And now let's look at whether the rash is smaller than or larger than 5 centimeters. And so here again, you can see it's imperfect, right? But here we have a larger proportion of people testing positive for Lyme in the lab that are presenting with that large rash. One more, please. And so if we go, here we go forward, yeah. So we go a level down. Here we sort people with and without the rash, and then maybe we go one level further and we sort people according to the size of the rash. But you see we're going to have some misclassification here too, right? Next slide, please. And so now I want to give you this challenge, and this is what physicians and clinicians are faced with, is that we have these individuals, and so what I would say is that this is very difficult, right? We might sort of average this person and say, on average, this is what we would expect. We would expect them to test positive or negative for Lyme, but it's difficult to get all the nuances here, right? And so this is difficult for us, but it's actually surprisingly easy for a machine learning algorithm. Next slide, please. So I'm going to give you an overview of the model, and you can just go ahead and advance through this. That's perfect. So we have 5 years of clinical survey data that went into this over 6 sites in 3 states. About 200 potential Lyme cases versus the endemic controls. And then here I'm sort of giving it away. I'm going to tell you the final numbers right here, the individuals who tested positive in the lab according to that 2-tiered testing, the serology and PCR. So 81 out of 297 tested positive and 216 negative. So with disease data, which I have worked with for the last 10 years, this is not uncommon. What we get is sort of this class imbalance. This is a very special problem for machine learning algorithms because it has to find a needle in a haystack. So we have 27% prevalence of individuals in this particular subset or mini population here. So we have to do some special things when we train our algorithm. And then there are about 50 features or traits from surveys that we talked about, so medication history, symptoms, et cetera, that I talked about in the first slide. Next slide, please. And so what we want, just one step back. So what we want to get out of the model, just to remind you again, is we want to predict or classify individuals as 0s or 1s, as Lyme negative or Lyme positive. And so what we actually get out of a model like a random forest, which is what I'm going to talk about, is very useful because we get a probability score. We can draw that line at 50% and say everybody above that is likely to be Lyme positive and everybody below that is likely to be Lyme negative. But we get a finer granularity. We get an actual probability value. And that means that if we want to be more or less conservative, we can classify individuals at a different threshold. Okay, so this is the most confusing and almost the last slide I'm going to show you here. This is actually aptly named a confusion matrix. And so I'm going to talk about the performance on the test set. Remember, this is just a subset of the data that we have held out. The model has not seen this in training. And we're going to ask how well does it classify these new samples that it has not yet seen. And so let's see if this works. Yay. Okay, so along the top here, this is lab confirmed. And then on the left are how the model predicted. And so I usually sort of animate this slowly so people can digest bits. So just look at the bottom here. And so you can see a whopping majority of these zeros, or people who were tested negative in the lab for Lyme, are identified by our model. So we're calling these true negatives simply because what the lab is telling us and what the model is predicting is the same thing. This is unsurprising. Remember, there are more zeros. It's an easier target for the model to see. The true challenge is whether it can get this smaller number. And so you can see we've done relatively well, I'll say here. So the agreement between what the lab is telling us and what the model is telling us is the majority, but not all of the individuals. So the goal really is to increase in this number, right? Ideally, we want to do perfectly here. Nothing is perfect, but we want to do as close to perfect as we can with this model. And a lot of that has to do with incorporating more data. And so really, we want to minimize false negatives and find everyone who is Lyme positive, everyone up here. And so what we do when we train our model is we optimize for something called recall. And that is finding all of the true positives, even at the expense of calling some people who are not Lyme positive. Lyme positive. And so one of the things in a tree-based algorithm that you get is an importance score. And the reason that I have this image here is because these feature importance scores. Don't worry about the magnitude of them, but they're where the splits happen. They tell you which things are at the top of the tree and allow you to make the first splits as you're sorting through all of these people. And so what you can see is this. And remember, this algorithm knows nothing. This is not an educated algorithm. This is a blind algorithm, picks out things that we know to be important. So the size of the rash, whether it's expanding, and then some of these symptoms appear very early, and that's telling you that's really what's helping the algorithm to do its job in classifying. So from here, really, it's all about collecting more data. That's the most important thing. These algorithms need more data to chew on. And if you're interested in hearing more about this, I would encourage you to talk to Liz, who is over here. She has 2 posters in the poster session, and she can tell you more about the great work that's being done by the Lyme Disease Biobank. So I would like to thank the Cohen Foundation and the Bay Area Lyme Foundation. And if we have time, I'd be happy to take questions. Sure.
Tests come out, would your biobank be able to basically upgrade your predictive algorithms for the new diagnostic tests?
Absolutely. So what you're talking about is if the target, or what we're looking for, that positive or negative, if that changes, yes, it's very easy to redefine what our target is or what we're looking for, that classification. So it would essentially be the same labels, 0 or 1, but the definition of that would change.
So you're using as the ground truth the serology?
Yes.
So what if that's wrong?
Yes. So I think
that's the problem.
Right.
I mean, that's the huge problem on all of this. But we had to pick something, so we went with positive two-tier serology. Oh, sorry. Yes. So the question was about ground truth being serology, and we know that two-tier serology is not perfect. We know it's not perfect in early disease, but that's what we used for this. But what is really interesting is it showed us some things that were important and some things that were less important. I don't know if you wanted to talk about that at all.
Important in context of the serology. But if serology is flawed, then whatever you're talking about is important is not actually valid.
Yeah. So I think, to that point and to what I have heard in this morning's sessions, I think we are looking at sort of a moving target because we're in a period of time when there's sort of exponential increase in the investment in trying to build new diagnostics. And so, yes, you know, we have something that right now is in an evolution, and as this changes, it will be very easy. Once the structure or the framework is built, it's very easy to change the input and see how that's reflected in the output. Yes. Yeah.
When you were building the predictive model, did you use a more like 1 to 5 sort of score for some of the traits, or was it easier to use like a binary, like yes or no?
Yep, so I chose to see this just as a classification problem. One of the big challenges, though, was dealing with the fact that we have, you know, many more zeros than ones. And so one of the approaches that I took was a method to generate synthetic samples. So there's a way to sort of use some nearest neighbor techniques to generate samples to balance the two groups out when you're training the algorithm. And then you take that and you apply it to the test set, which has been held out, that has that bias, and see how well it does. But it actually helps in the training of the algorithm to be able to see those two groups a little bit more clearly.
Please help me in thanking Dr. Gervaisi. Okay, so for our next, we have 2 presenters. They're going to co-present, and they're research scientists from UCLA. And they're going to present point-of-care Lyme serodiagnostic test using a multiplexed vertical flow assay and machine learning. Please help me in welcome Zachary Ballard and Hyou-Arm Joung.
I thought I had the magic clicker, but this is a replacement clicker, but I guess this one's not working either. Yeah, it's on. Could you just, oh, looks like we have another replacement here. Total clicker failure. Yeah. I'll just verbally. All right, sorry about the technical difficulties, everybody. Could you advance the slide to the title? Thank you so much. So my name is Zachary Ballard. I am a senior PhD student. Actually, will be a doctor in about 3 days, so very excited about that. From UCLA. And
I'm the Dr. Hyou-Arm Joung and postdoctoral scholar at the same laboratory with Jack.
Yeah, and it's a great privilege and pleasure to be here to share with you some of our really exciting results with our diagnostic platform that we've been working on in the lab. And before we start, we'd really like to thank the Cohen Foundation, as they've been supporting us through this research effort and been wonderful partners, as well as the Lyme Biobank. So you can go ahead to the next slide. Thank you. Just a brief introduction to the team. So we work with 3 different researchers at UCLA. Professor Idawan Ozcan of the Electrical Engineering Department, Professor Dino DiCarlo is in bioengineering, and then we work with a physician, Dr. Amai Garner, who's actually the head of point-of-care testing for the UCLA health system. We go to the next slide. Right, so our lab is focused on bringing diagnostics to the point of care. So this means making a diagnostic test that is low cost, that can be performed in an office setting, and that can get results rapidly, so in a 15 to 20 minute time window. And it's really to address these pain points that you all might be familiar with current serological testing, specifically the 2-tier method. So the 2-tier method has very high cost per test. As we heard this morning from Anne-Marie, this can actually make it prohibitive to people who are uninsured from getting testing. It also has a slow turnaround time, as samples have to be sent to a centralized facility where trained technicians using benchtop instruments run tests and then send results back. And one other major pain point of current Lyme diagnostics is very poor sensitivity for early stage samples, which is the time when patients often go to seek treatment. So on this slide is an overview of our proposed system. And what it is actually a paper-based test. And I actually have it here, so it's this size. It's really just made of paper and 3D printed plastic. And we have a mobile phone reader that can actually take an image of this test after the assay is complete, and automated computational processing to actually output a seropositive or seronegative diagnosis through automated image processing that's done either on the mobile phone or a remote server. And to try to combat the poor sensitivity in early stage Lyme, we focused heavily on making this a multi-antigen test. So this test actually detects the presence of multiple Lyme-specific antibodies in the blood in a single go of the assay. So go ahead to the next slide. So the way this test works, actually, it's the chemistry within this cassette is identical to that of a lateral flow assay, which you all might know of as a common pregnancy test. And it's a paper-based test where blood or serum is wicked across a strip of paper, and the antibodies in that serum are immobilized on a strip. And then there's typically a conjugate pad, which is shown there in the yellow, and this contains gold nanoparticles that go and tag the immobilized antibodies. And this gives that dark pink color that would indicate the presence of antibodies or not. And this is a really ubiquitous technology that's been in point-of-care diagnostics for many years. However, it has an inline geometry which prevents multiple antibodies from being measured in a single test. So if you go ahead and click next. So what Hyou-Arm and I have been working on at UCLA is developing a vertical flow assay that works similarly to the lateral flow assay, except that blood is driven through a stack of paper layers and can actually interact with a two-dimensional sensing membrane, which is shown there with the underlined red, the multiplex sensing membrane. And go ahead to the next slide. And so this is a zoom in of the multiplex sensing membrane. And it has a number of different spots here, each of which are responsible for creating a signal that corresponds to the concentration of Lyme-specific antibodies in your blood. So that's what's shown in this cartoon here. And we can, with this test, we can measure up to 13 Lyme-specific antibodies in a single assay. And it also opens the door for detecting other tick-borne illnesses, again, with the same single test. Go ahead to the next slide, please. The operation of the assay is very simple. We've really tried to limit ourselves so that this could be performed in an office setting or urgent care clinic. So there's two parts to the operation. The first part is just the introduction of some buffers, as well as a serum sample, and then a passive incubation step. And then after the first part of the assay, the top of the cassette is removed. And you go ahead to the next part. And a second top case is then twisted onto the cassette. And this second top case contains those gold nanoparticles that create the color signal. So that's what we see here. And we've actually created two assays, one that detects IgM class antibodies and one that detects IgG class antibodies. And these, of course, can be run in parallel, as a lot of the assay time is just idle time and passive incubation. And I'm going to actually have Hyou-Arm pass this around so you guys can play with it and get a close look of it, just because it's a really awesome platform we're really excited about. Go ahead to the next slide, please. So after the assay is complete, we have a mobile phone reader that we've developed. So this is, again, a very low-cost piece of technology. It's made from 3D printed plastic. We have 4 different LEDs that illuminate the multiplex sensing membrane, and then an external lens that focuses on the sensing membrane to take a picture. The material costs of the reader are about $20. It's battery powered, it's handheld, it's not a benchtop reader, it shouldn't be taking up much space in an office or clinic. Go ahead to the next slide, please. And after the image is taken, we have some computational processing that extracts the pixel intensity of the different reaction spots. And each pixel intensity corresponds to a different Lyme-specific antibody that are shown in that color-coded map. And then all of these different signals are fed into a neural network for inferring a positive or negative diagnosis. And I really appreciate Stephanie for giving everyone a really great introduction to machine learning. We are using machine learning here, except with clinical symptoms, we're just looking at the antibody signals coming from our test. Go ahead to the next slide, please. And we've had the great privilege of working with the Lyme Disease Biobank to do a machine learning study with this platform. And the way the study worked, it was broken up into two parts. The first was running our test to create a training data set. That's what's shown on this slide. So we actually were able to run our tests with 50 human serum samples, 25 of which were Lyme positive, 25 of which were endemic controls. And we ran these tests in duplicate, which means we actually activated in the lab 100 of the IgM cassettes and 100 of the IgG cassettes. And that's what's shown on this slide here. It's a lot of busy data, but essentially the red indicates a positive presence of that Lyme-specific antibody that's shown on the x-axis of the graphs there. And these are all the positive samples. So there's a lot of information here that the machine learning and neural network helps us harness to infer a final diagnosis. Go ahead to the next slide, please. And another task that we performed with this training data set was actually to try to select a subset of all of the different Lyme-specific antibodies to analyze for a final diagnosis. In fact, looking at a smaller subset of the antibodies can give us improved diagnostic performance, which is what this graph shows here. So there's a lot of details here, but essentially the AUC is a measure of the diagnostic performance. It stands for the area under the curve of the receiver-operator characteristics, so that's shown on the left. We have the confusion matrix that Stephanie explained earlier, shown in the inset. And essentially, diagnostic performance can improve as we add different antibody signals to our analysis. And then eventually, if you add too many antibody signals, some of them contain noise, so the diagnostic performance can decline. So this just graph shows that there is an optimal performance with a given subset. And again, we found this using the training data provided by the Lyme Biobank and run on our assay. And after we determined this subset, we trained our neural network to infer a seropositive or negative diagnosis. And then we received a fully blinded panel from the Lyme Biobank and performed a blinded clinical study. So go to the next slide, please. Before I move on, this is the subset of antibodies we're using. So there's actually 6 antigen targets we've incorporated across the IgM and IgG assay cassettes. And then we have some positive and negative control spots as well. And this, again, improves performance, but also can lower costs. And now we know that the antibody signals that are not useful, they can maybe be replaced with redundancies of the useful information, or maybe new antigen targets that can indicate other tick-borne illnesses. So go to the next slide, please. So with the blinded study, we just completed this and have some very exciting results. We achieved a sensitivity of around 90% and a specificity around 87%. However, there's some decision threshold engineering that can occur to be more conservative in terms of the specificity, so lowering the number of false positives. Those results are shown in the parentheses there, 87.5% sensitivity, so a little bit lower sensitivity at the cost of a higher specificity, being more conservative to false positives. So we're really excited about these blind testing results. Again, this is an assay. The assay cassette costs under $1. It can be run in 15 minutes. With a clinician who's trained in maybe 10 or 15 minutes, and can be run in an office setting. And you go to the next slide, please. And we really look forward to pushing this technology forward. We would love to see it eventually in the market. And we're looking to combine the IgM and IgG assay into a single cassette, and then perform larger clinical studies that, with more meaningful, larger data sets across different geographical areas and with a larger, more diverse sample population. So with that, I'd like to thank you, and I'll hand it over to Hyou-Arm just for some final words before some questions.
Yeah. Thank you for your attention. And actually, I'm the researcher who have been contributed over more than 15 years for the development of point-of-care sensor platforms during the industry and academia. So I'm sure that this platform is ready for the fighting at the real field for the Lyme disease. Thank you for your attention.
Thank you. Yeah, a couple questions.
I don't need a microphone. All right, I'll use the microphone.
You're still going after the antibodies, and therefore you have the same problem of people coming in, having Lyme disease, but not developed the antibodies yet, and you're going to come up negative.
This is absolutely true. It's the exact same problem that we're having. There is no good gold standard. Now, because of the well-characterized samples from the biobank, we can actually make a gold standard that combines the serological testing with PCR. So someone who's maybe seronegative but positive in the PCR can be a gold standard. So we capture some of those samples, but it's a fundamental problem. Absolutely, and it's, unfortunately, there's no immediate solution for us. We just want to increase the sensitivity of two-tier testing in a cheaper and faster way, and we think that that can have great impact, expanding the access to this testing. But it's an excellent question and point.
I think there's a lot more questions, but they're going to have to be saved for after the next discussion. We're running out of time here, so thank you. Help me thank the presenters. Thank you very much.
Thank you.
Okay, so we're going to move on to the next session. Let's see here. This is a lab talk. So we had multiple proposals, abstracts submitted, all from the same lab. And so we said, why don't we just invite the whole lab here to give a lab talk? And so this is Errol Fickred's lab. Why don't you join me up here? And I think, I have a textbook description of what you do, but you're here. Why don't you tell us about your lab?
Hello. My name's Errol Fickred. I've worked on Lyme disease for quite some time. I won't bore you with my lab. Let's go to the hot stuff, what's going on today. Yeah. But one of the great pleasures in life is to help train the next generation of researchers for tick-borne diseases, and we have 3 here for you today. Shane Kurakawa, Aurobind Vidyarthi, and Jeff Lynn, and they'll be telling you about the work they're doing to develop a new vaccine to prevent tick-borne diseases. So we have a next slide. I've got one slide. Can we? Yeah. There's a slide guy somewhere?
We were going to take a 2-minute break
Ah.
this, and I totally forgot that.
Okay. I can sing or dance? No, you really don't want to see that. There we go. We've got a slide. Okay. So in simple terms, our main goal is to change a tick bite into a mosquito bite. So think about it. When you get a tick bite, what do? You probably haven't noticed it for a while. First, you get disgust, right? And then you search around quickly for a pair of tweezers, and you spend some time plucking it out. But it's silent. Nobody feels the pain of a tick bite. That's unusual. What's your response to a mosquito bite? Right? It's quick. So our goal is to make it so that when a tick bites you, you get that similar sensation. So what you'll do is very quickly, before the tick has started feeding, you'll pluck it off. So this is how that works in the laboratory. On the top, there's a cartoon of a tick feeding on, and this is a guinea pig. And there are a bunch of, I would call them happy ticks. And you can see in little black spots, they're feeding properly on a guinea pig if you're a tick. Now, if you put 50 ticks in a guinea pig and let them feed, they'll all feed happily. If you let those ticks feed and come back about 2 weeks or a month later and put another 50 ticks in the guinea pig, you get this happening. The ticks, you see it in gray there, it inserts its hypostome, it doesn't feed well, a lot of redness develops at the tick bite, the ticks fall off, and for the most part, they die. So we're trying to determine what are the mechanisms by what that phenomenon called tick immunity occurs, and identify the antigens that cause it, and hopefully develop a vaccine that makes a tick bite become more like a mosquito bite. So they'll tell you all about the work. This work was actually jump-started by Jennifer Monsky, her support in the laboratory, and then later on, Jennifer's here, and later on now by the Cohen Foundation and by the National Institute of Health. So I'll give the stage to Shane to start.
Okay, can we just get the next slide? All right, so thank you all for having us here and letting our lab kind of tell you about the work that we're doing to try and make an anti-tick vaccine. Next slide, please. So as everyone's aware, incidences of Lyme disease have really been increasing over the past 20 years or so. And Lyme disease, of course, is caused by Borrelia burgdorferi, which is transmitted by the Ixodes tick, Ixodes scapularis tick, mostly in the US. And despite years of research about Borrelia as well as efforts for a vaccine, there currently is still no vaccine available, effective vaccine. Next slide, please. So shown here are several Borrelia proteins that have been targeted for vaccine development. I'm not going to go through every single one on this list, but I just want to point out that these proteins that have been targeted, they all have essential functions for Borrelia in its pathogenesis, making them ideal vaccine candidates. However, there have been several hurdles preventing the successful development of these as vaccines. For example, for a lot of the outer membrane surface proteins, there's variable expression during the life stage, dependent on the life stage of the Borrelia, where it's at in its kind of cycle of pathogenesis. Similarly, there's a lot of heterogeneity for a lot of these outer surface proteins among the different strains. For example, with the OSP-C proteins, the protein can vary among the different strains, making it difficult for a single vaccine to provide broad spectrum protection against the different type of strains out there. And another example of this is with the VLS-E protein. And you have to keep in mind that these pathogens have evolved very complex strategies to avoid neutralization from their host. And a good example of that is with this VLS-E protein that has a very complicated system of recombination that allows it to have continuously alter its antigenic properties, making it very difficult to target. So overall, targeting Borrelia proteins can be quite difficult and has not been as successful as we need to control Borrelia. Can we get the next slide, please? So what our lab is doing is we're looking to see if we can actually target tick proteins rather than the pathogens that they transmit, with the idea being that if we could attack some of these tick proteins and induce an immunological response towards the tick, that could inhibit or disrupt tick feeding. And in the process, you would thus prevent pathogen transmission, the pathogens that these ticks are transmitting. Next slide, please. So that's what we're going to talk about today. And just before I get into that, a quick overview of how this talk will go is I'll give you a background on this tick immunity, a little bit more background, and some of the work that our lab is doing, specifically trying to identify a source and a protective antigen for a vaccine. And then the second part of the talk will be Arben. He's going to be talking kind of about the immune mechanisms and this phenomenon of where mice do not develop tick immunity and see if we can learn anything from that. And then we'll close the session with Jeff, who's also a postdoc in the lab, and he will talk about if these vaccine candidates can provide broad cross-protection for other tick species, as well as looking at other sources of tick antigens where other than salivary proteins that I'll get into. So next slide, please. So this concept of tick immunity has actually been, the observations have been around for quite some time. These were actually made for the first time by Bill Traeger back in 1939. And what he observed was an immunologically acquired resistance to tick bites in animals following repeated tick feedings. So this kind of set the tone and the idea that maybe we could target tick proteins rather than the pathogens they transmit. And more specifically, what Dr. Traeger recognized was that if you took ticks and kept putting them on the animal, what would happen was you would have rapid tick detachment of the new ticks. These ticks that did attach and then detached rapidly, they were not able to engorge and acquire a full blood meal. Additionally, these ticks did not molt, so they did not go from the nymph larvae to nymph and nymph to adult stage. And additionally, at all of the new bite sites, there was erythema at these bite sites, characterized by redness at the bite site. And this was observed for several species of tick, Dermacentor, Amblyoma, and so forth. And while this concept has been around for quite some time, the specific mechanism of rejection and the antigens targeted remain unknown. Next slide, please. And there is some evidence that an anti-tick vaccine could potentially work in humans. So here was a study where they immunized cattle with a Borrelia protein. And what these authors of this study observed was, yes, of tick proteins rather. And what they observed was that the ticks became partially resistant to tick feeding. Next slide. So the model that we're working with is an Ixodes model, obviously because it transmits Borrelia. It's the primary vector, and it's also a vector for Babesia, Anaplasma, and Powassan virus. Next slide, please. So what this looks like in our guinea pig model is that when you apply ticks for the first time on the guinea pig, you will see this is a normal tick feeding pattern where you see that the ticks attach, and then around 48 to 72 hours, they're starting to complete the feeding, and they start to detach and release from the guinea pig. And then we recover those ticks, and we weigh them to assess how efficient they were able to engorge. And as you can see, they weigh around 3 milligrams. Next slide, please. When you come back 2 to 3 weeks later and apply ticks for the second time, you can see that the rate of detachment starts to occur much faster, where you see that they're almost fully detached by 72 hours. And then when you look at their engorgement weights, you can see that only a few of the ticks were able to fully engorge, even though we put the same number of ticks. And next slide, please. And when you come back for the third time, you can see that the ticks rapidly detach, where they're almost completely gone by 48 hours, and none of the ticks were able to fully engorge. So it's quite impressive. Next slide. And if you look at the bite sites of these new feedings, you can see that by the second tick exposure, there's already erythema at the tick bite sites. There's this redness at the bite site, suggesting that there's an immune infiltration to the bite sites in an antigen-specific manner. However, these protective antigens remain unknown. Next slide. So in order to try and identify a pool of antigens that could possibly serve as our vaccine candidate, we examined the tick-host interface, and we wanted to look at how that was happening, because when a tick feeds, it really needs to accomplish several goals. It needs to increase the blood flow, and it also needs to suppress the immune system to prevent the host from really detecting the host, kind of what Dr. Fickrick was talking about when we want to make sure that you can feel it. So next slide, please. A lot of work has been done in the tick saliva to try and identify what proteins are in there. And there's hundreds of proteins in the tick saliva. And as far as assessing the function, shown here is just a brief summary of these functions of these proteins in tick saliva, where you can see that they play a role in vasodilation, wound healing, as well as controlling the innate immune response. Next slide, please. And even probably more impressive and more important is that these tick proteins have also been shown to play an essential role in enhancing the spread of Borrelia transmission. So this was work done by our lab a few years ago, where they demonstrated that SALT15 was actually able to enhance transmission. Next slide, please. So based on this, we hypothesized that vaccines targeting critical salivary proteins can prevent successful tick feeding, resulting in tick rejection. And by targeting these tick proteins, this strategy can be utilized to prevent the spread of several tick-borne pathogens, such as Borrelia burgdorferi. Next slide. So in order to test this, what we wanted to do was we immunized the animals every 2 weeks for a total of 3 doses, and then we measured their serum response 2 weeks after the final boost. And what we saw was that these guinea pigs could successfully be immunized by ELISA, where we can detect saliva-specific antibodies. Next slide. So following up on that, the next thing was to challenge these immunized animals with ticks. And as you can see, we are able to observe some protection in the saliva immunized group relative to the control group, where you can see a rapid detachment and significant detachment of these ticks relative to the control. Next slide, please. And when we compare that with the tick immune animals, we can see that they're also able to engorge less, similar to the tick immune animals, but much lower than the ova control animals. And similarly, there's increased redness at the tick bite sites that was not observed in the ova control group. Next slide. And when we look histologically at these bite sites, what we see is that there's a significant infiltration of immune cells specifically towards the bite sites. And we can see that in the PBS control group relative to the saliva group, where there's a robust influx of eosinophils, mononuclear cells, such as T and B cells, and mast cells as well. Next slide, please. And following up on these results, we can see that saliva is able to provide some protection, but we were also interested in trying to identify the factors in saliva that mediate this protection. So in order to do this, we wanted to assess the role of proteins, sugars, glycoproteins, lipidated proteins, as well as phosphorylated proteins. So we took saliva and we treated it with the various enzymes, a protease, a glycosidase, and so forth, and then used that as, as the immunizing antigen, and we followed the same immunizing protocol. And as you can see, the PBS group, the ticks are able to feed efficiently. In the saliva group, as I showed before, the ticks are not able to feed efficiently. However, when you eliminate the proteins or eliminate the sugars, you rescue the phenotype, and the ticks are now able to feed, suggesting that the proteins and these glycosylation patterns are critical for eliciting tick immunity. Conversely, lipase and phosphatase treatment had no effect on the overall impact of immunizing these animals and providing protection. Next slide. So kind of the next steps moving forward are trying to identify the proteins in saliva that are important for providing protection. We know it's a glycosylated protein that seems to be important. So work we've done in the past is we've taken saliva and probed it with tick immune serum and have identified several proteins. Shown here is just a list of 14 or so proteins that have been shown to react with guinea pig serum and could be possible in providing protection. So we're in the process of kind of cloning all of these and immunizing guinea pigs to see which one provides protection. Next slide. So in conclusion, we've shown that saliva immunization elicits protection against tick bites, and upon tick challenge, these immunized animals elicit a memory response towards the new tick bites, and that glycosylated proteins in saliva are successful for vaccination. And you know, with the screen of about 14 proteins, we hope to continue the screening process and identify the essential proteins. Next slide. Yeah, I kind of covered that, and I think I'm running out of time. So next slide. I just want to acknowledge everyone in the lab and, of course, the Cohen Foundation for the funding. Thank you. Yeah.
We're going to save all questions to the end, time permitting, so everybody gets a chance to present. Thank you so much,
Yep.
Shane. And then next, is it Jeffrey or Arvin? Arvin. Oh, hey.
Yeah,
So
good.
this is the placebo.
Okay, I will
for
try
you.
again. Okay, no worries. Oh, yeah, it's working. Oh, no, that's not. I maybe.
Fooled by the human.
Oh, cool, it's working. So, first of all, thank you for the introduction, and I would like to thank Lyme for giving this opportunity to share our data with you people. So, you all now know, because Shane and I all told about tick immunity, so it exists, and it is known since long back. But we are not able to develop a vaccine, so our lab is screening some candidates, but the second most important reason that why we are not able to develop tick vaccine is this, because we have insufficient knowledge about the immune responses. The main reason is because we don't understand when there's tick immunity in guinea pigs, that how those guinea pigs are getting immune. What are the immune mechanisms? What are the immune cell infiltration in those guinea pigs? We don't understand that mechanism. So it's very important to understand the mechanism that how this tick immunity is inducing. So this will be a little bit repeating, but I just will focus on main points. So this is the life cycle of Zytus Capillaris, which is a tick which is responsible for Borrelia transmission, and that causes Lyme disease. So there are different stages of tick development, from eggs to larva and adult. And during these stages, these ticks fed for blood meal on different hosts, like humans, deers, and mice. So I will be concentrating on mice, and I will discuss the reasons why. Oh, it stopped working. Next slide, please. So these are the two main reasons we wanted to study tick immunity in mice. One of the first reason, which I already discussed, it is a natural host for Zytus Scapularis. And second, if you want to study the immune responses, there are a lot of tools available and genetic knockouts are available for mice. So you can study that in mice. I want to discuss one study here in which they have talked about like non-permissive host, like guinea pigs, the way we have showed they have tick immunity. And they have talked about permissive host, like mice. And we think why this mice is a permissive host, because during evolution, because mice is a natural host, so they have co-evolved. So ticks and mice are co-evolved naturally, and that's why they are not able to get tick immunity in mice. So they have shown the only one thing in this study is like there is a difference in the lesion architecture. When ticks bite the mice, there is only the difference in lesion architecture of these two, permissive and non-permissive host. But we don't understand the mechanisms, how it's working. So to ask this question, next slide, please. We followed the same strategies as Shane has shown. I will just be briefly sharing what we do. We took mice and we do tick challenge, and after every 2 weeks we do tick challenge and third tick challenge, and then we measure tick attachment. If you see the left side of the graph, after first, second, and third tick attachment, it is exactly opposite to the guinea pig. You don't see tick immunity in mice. And to confirm that, we checked the tick weights, and if you see the tick engagement, there is no difference in the tick weight of first, second, and third exposure. To further confirm our experiments, we also done saliva immunization. Next slide, please. So we confirmed these experiments, the similar data we get with saliva also. So when you give saliva, immunize with saliva, the mice, you don't see a difference in the tick fall rate. And if you see in the right side panel, there is no difference in the tick weight. Next slide, please. So the main question we wanted to ask, okay, what is the immune response differences between guinea pigs and mice? So first thing we asked whether there is antibody generation in mice. So we took saliva and we checked for the antibodies against saliva after first exposure and second exposure. From the serum we isolated from the mice. And clearly you can see that there is antibody responses in mice against saliva proteins. We are doing some experiments and where we are able to find that these antibody differences are to different proteins. Maybe they are not protective in mice and they're protective in guinea pigs. Right now I'm not showing that data, but we are getting it. It's very preliminary. Next slide, please. So after seeing, okay, there is antibody responses, so we wanted to check what about the cellular responses in mice. So on the left side panel, if you see, we took the draining lymph nodes from the naive mice and tick-challenged mice. And when we checked for T cell infiltration, CD4 and CD8, and on the right side, we have shown the bar diagram, the same data. Clearly, there is massive infiltration of cells in the draining lymph node. So the basic idea is this, like there is an immune response to saliva immunization and ticks in mice, but that is not protective. So I just want to conclude this. Next slide. So the two conclusions which I talked first, that repeated tick infestation and saliva immunization does not induce tick immunity. And second, the most important thing is that immune response is there, but it is a differential in differential in mice and guinea pigs. So next slide, please. So the idea of this project is we wanted to compare mice and guinea pigs' immune responses, and that will give us hopefully insights into the mechanism, and that mechanism will definitely help us to develop a better vaccine so that we can decide what adjuvants to use for that vaccine if we know the mechanism. Thank you very much. If you have any questions. Last slide, please. I just want to thank Joe Kraft and Errol Fickrig. They are incredible mentors, and most of the work I have done with Shane, whatever I have shown, and definitely without funding, nothing is possible. The Cohen Foundation. Thank you very much.
We'll save the questions for the end.
Do that?
Yes.
Is that
And that'll
still
leave
working?
this, yeah. So
Sometimes? Which one?
I
Okay.
think you just tell them next
Okay.
slide.
Okay. All right. So I'm going to pick up where Shane left off a little bit in terms of screening tick tissues and extracts for potential vaccine candidates. So salivary glands and saliva have been studied quite a bit for many tick species for clear reasons, but there are also several other tissues, midgut and cement, that have had some degrees of success in terms of immunizing animals against ticks previously. So I'm going to talk about that a bit. Again, this is a black-legged tick. It's our primary concern because it is the main vector of Lyme, the pathogen that causes Lyme disease and a number of other important tick-borne diseases in the US. And so for this, again, there has been a lot of good work done with saliva and salivary glands, not nearly as much with midgut and cement. So that's something that we wanted to look into. And so just briefly, for those of you who are not familiar with tick anatomy, this is the midgut of an Zytus scapularis. It's a very large organ, so there are lots of potential antigens to look at there. And it also has the advantage of, at least in some cases, for other tick species containing both concealed and exposed antigens. And so if you're able to identify one of those, it has the potential to have a dual effect where there's both a host response at the site of feeding as well as possibly an internal effect on, say, the tick midgut, like that's the mechanism for the BM86 cattle vaccine. So this is the first vaccine trial then that I did with midgut extract. It's a similar vaccination schedule to the work we were doing with saliva. So I was using this extract to immunize guinea pigs at 2-week intervals, followed by a challenge with Scapularis nymphs. So what you can see there is the tick weights from the ticks from each of these animals. And you can see there's really no difference in tick weights between the control and the two immunized, the ticks from the immunized guinea pigs. So I've got some ideas on why that was, but we don't have time to get into that today, unfortunately. So the second substance then is that I'm interested in is tick cement. So tick cement is very important for the tick in terms of attaching its mouth parts to the host. It's also supposed to help in the process of feeding and is hypothesized to have to help protect host mouth parts and against, the tick mouth parts against the host's immune response and potentially harmful substances. So I've got a few slides here just to give you a better idea of what tick cement looks like and how it interacts with the host. So on the right, there is a cross-section of a tick feeding embedded in the host skin. On the right, a higher magnification shot that I think shows well the sort of interface between those mouth parts, the feeding lesion, and that's where you're going to find a lot of the cement. Again, a couple more shots. So different tick species have different amounts and really types of cement. So this one is a nice example. You can see the pink along the top. A lot of that is, there's a lot of cement in that case. We've got a nice diagram to the right that shows both these kind of lateral projections of cement as well as the case around the mouth parts and even how it sort of perforates into the host skin with these projections. So there's a lot of different compounds within tick cement, but so far, at least from some of the other species that we've looked at, our other labs have the keratin-like and collagen-like proteins seem to be good candidates for vaccines. So again, same vaccination strategy here with cement extract. And so we were able to see a result here. There was, in 2 guinea pigs vaccinated with cement extract, there was between a 35 and 41% reduction in tick engorgement weights relative to the control. Was a significant difference, and we're still waiting to see what for the ticks to molt, but I'm guessing somewhere between a third and half the ticks there that fed on the 2 cement guinea pigs will not survive and molt onto the next stage. So it's not ideal in terms of 100% rejection. It's not nearly as good as the saliva, but this is part of the screening process. We don't really know what's in the mixture of cement yet, and so if we're able to identify specific components of it and combine those with other effective proteins, increase the concentration in our vaccine, they could potentially there could be a much greater effect. So the last topic I wanted to cover briefly was just the concept of cross-species tick protection. And that is basically the idea is that protection or resistance against one tick species can be conferred to a secondary tick species. So this has actually been demonstrated in the past for non-Exodes species, at least starting with them. There's also been some work showing cross-reactivity between animals that are resistant, the serum of animals that are resistant to a specific kind of tick against antigens from other kinds of ticks. And importantly, the example of the P64 protein from Pat Nuttall's lab that actually conferred resistance against 4 different species that were not, different species from the type the protein originated from. Also showed cross-reactivity against them. So we're going to look at this further, starting with the Exodes scapularis proteins. And the idea, again, is that if to come up with a vaccine that is broadly applicable not only to Exodes scapularis, but to some of the secondary vectors of disease in the US. So that's it. To acknowledge the other members of the FICRG lab, Joe Kraft's lab, and our funders, the Cohen Foundation.
So we have 5 minutes
Sure.
for Q&A. So there were a lot of questions.
Can we reverse it and first I ask the audience the question?
Yes, absolutely.
Okay. So we're tired of answering questions. No, happy to answer them. So the general question really for the general public is, if there was a vaccine, but it required a little bit of redness at a bite site, would you want to take it?
Yes. If it was an anti-tick vaccine?
Anti-tick vaccine, but okay, because I'm not sure, I actually was not sure about that, and I'm not sure how the public would respond to having a vaccine that worked to prevent tick-borne diseases but required redness at the tick bite site. I still think that's a bit of an open question, but I'm glad to see some support. Okay, it's good. It's good to hear. But any questions? Yes. Yes.
So humans are a dead end in the cycle.
Yep.
Humans are a dead end in the cycle of disease, whereas the reservoir animals are not.
Yep.
I've welcomed a tick vaccine since I first met Stephen Weichel about 20 years ago, and so I compliment you for tackling this. I'm very excited. The problem is that I see is that the mice, it doesn't affect them, and they're one of the prime reservoirs. Do you think there's any way that we could get some kind of bait vaccine for the mice that would immunize them, the mice and other reservoir animals? Because we really need to knock down the tick population and prevent them from being able to transmit pathogens.
Yeah, that's a very good question. I view it as a bit of a separate question because I don't think you can actually do that with an anti-tick vaccine. I agree with you completely. There are mice, like Aurobind showed the data in mice. So the question is, first question is, are we more like mice or pigs? I personally think, anecdotally first, I think we're more like pigs, and I say that because if you talk to older residents in Block Island, and you ask them about whether they care that much about Lyme disease, the general answer is no, because they tell you that ticks don't bother them anymore. Whether that's, there's no scientific data to show that people get tick immunity, but my guess from what I've heard anecdotally is they do, okay? But the second question, how to prevent Lyme disease in the reservoir population. That's more likely going to be addressed by targeting perhaps borrelial targets rather than tick targets. I would find it, ticks and mice have co-evolved for for millions of years. I think it's a very intimate relationship. Understanding why a mouse does not get tick immunity may lead us to understand why we do. That's why we're doing the mouse work.
So
coevolution, yes.
We've got a bunch of questions.
Sorry,
So what I'm going to do is I'm going to ask that I'm going to ask people to frame their question with start with how, why, what, why, frame it as a quick question. We're going to do 3 in a row.
Okay, apologize. You take care of the speed. I'll blabber on and on, so you take care of this.
So sorry if I missed this at the beginning, but I wonder, does the life stage of the tick matter for the efficacy of the immunization property? Like, for instance, the first bite, if it's a nymph, does it protect against an adult or only another nymph?
Great question. We don't know the full answer of that. To separate it into 2 parts. The first one, the one you care most about is the nymph, because that's what causes most of the disease. The adult is also important. The larvae is not very important because Borrelia do not go from the tick. Larvae acquire the spirochete. So we don't really care that much about larval ticks. But short answer is yes, there is some degree of cross-protection. Protection against nymphal tick bite and adult tick bite has some cross-protectivity. How much it is, I can't tell you for sure yet. Very important question.
Hi, I'm really excited by your research. 2 questions. One follow-up from your question, which is in your data you saw that it's usually the 3rd tick bite in the guinea pig that gives them the immunity. So why is it that you need the 3rd tick bite? Is it something with the stage of the life cycle, or is it just that you need multiple exposures? Number 2 question is...
You're only allowed 1 question.
Sorry.
Go on, answer that question. My memory's not that good. No, so I'm stopping you now. 1 question. So it's an important question, but it's our cartoon that we do 3 tick bites over several periods of time. That's our standard model. We've adjusted that. And you can get tick immunity by one bout of tick feeding as well, if you wait a certain amount of time. So it's the importance of the immune response to tick saliva that occurs over time. So that 3 feedings is not written in stone. There are other ways to do that.
Got it. Thank you. And this is an important question I want
You're
to
giving
ask.
him a second question. Okay.
Largely because I've studied the history of the last Lyme vaccine.
Okay, get to the question.
The question is, how do you make sure that the saliva that you're extracting from the ticks are free from the Lyme bacteria?
So we have 2 things. First of all, at the end of the day, we're going to have a recombinant protein made with recombinant techniques in cells. So there's 0 association with Borrelia. As we do our studies, we have a laboratory colony of ticks. So we have the larvae, nymph, and adult stages feeding, and they feed on clean mice. So our laboratory population, we have a clean population of ticks, laboratory ticks without Borrelia, and laboratory ticks with Borrelia.
All right, wonderful piece of work. I'm looking forward to continue hearing about that. So this anti-tick vaccine will work against like Borrelia or any bacteria, but what about the viruses? They are readily delivered right there.
Great question. So every tick-borne pathogen is different. In general, Borrelia and Babesia take a long time to go from a tick to a mouse. So my guess is that if we're fortunate enough, this vaccine approach will work against Borrelia and Babesia. Anaplasma takes about 12 hours to go to a tick, from tick to mouse. Whether it's going to be good enough to prevent anaplasma infection, I don't know. And Powassan is very, very fast. So this has to be super effective to work against Powassan virus. So my initial bias is this would be unlikely to be effective against Powassan virus. And anaplasma, I can't say yet. More likely, no.
Let's thank the speakers. Okay, so what's next is there is a connectivity lunch, I think, happening in here. And there's also other activities happening throughout the building, and please check the program. And thank you for coming.
My name is Brooke Stoddard. I'm the co-founder of Generation Lyme, and we are an initiative of Project Lyme, which is a nonprofit based here in New York City that raises awareness of Lyme disease across the country. And specifically, Generation Lyme is a community of young people with Lyme disease. I started Generation Lyme with 5 friends across the country, all of whom have Lyme disease, because as we were going through our own personal health journeys, At times, we found the process to be very challenging and frankly, very isolating. And so the purpose of Generation Lyme is to reach out to young people and to help them feel less alone as they're going through their own personal health battles. So we do two things in Generation Lyme, and it's very simple. First, we manage an Instagram account, which you are more than welcome to follow. It's at @generation_Lyme. And on this account, we reach out to young Lyme disease patients across the country. We interview them. We have them send us a picture, and we tell their stories on Instagram. That means that anyone around the country can see these inspiring stories on our Instagram account and feel less alone as they're going through their own health battles. The second thing that we do is we host in-person meetups in New York, New Jersey, San Francisco, and Denver. And we also host conference call meetups for people who aren't able to attend in-person meetups in any of those areas. So if you have any friends, children, patients, neighbors who are young and would like to be a part of a community like this, I would encourage you to ask them to just follow us on Instagram at @generation_Lyme, or send me an email at generationlyme@projectlyme.org. Thank you so much, and now I'll hand things over to Evan Golub.
Thanks, Brooke. Left for Brooke. Thanks, Brooke. In January of 2013, I woke up with a horrible case of vertigo and disequilibrium, which progressed into 6 years of chronic illness. I was misdiagnosed for 4 years across 30 practitioners. It didn't make any sense. Before this, I'd barely taken as much as a Tylenol for a headache. Please raise your hand if you've experienced any of the following: migraines, vertigo, brain fog, light sensitivity, or memory loss. For those of you who have, you've probably felt frustrated, alone, and helpless. I'm Evan Golub, and I'm the co-founder and CEO of WANA, which stands for We Are Not Alone. WANA is a digital platform to serve the more than 30 million Americans who currently face invisible, modern, and chronic conditions, which are misunderstood and often misdiagnosed for years. It was my own health journey that led me to being here today. Through years of helplessness and frustration, I realized there was no centralized place to find others like me. With WANA, we've created a community where people can connect with one another, share their experiences, and find the right information and resources. We want to create a positive space where like-minded people can discuss their health journeys. We match members with similar symptoms and diagnoses so they can lean on one another. Community is good medicine, and the first step to healing is a strong connection. If you're looking for information on treatments such as acupuncture or cranial sacral therapy, or even helpful supplements such as glutathione or phosphatidylcholine, or you want to connect with others who can relate to you, then I encourage you to join our growing community by downloading the app. Healing is something you no longer have to do alone. Thank you so much. I hope you're all feeling well today. Thanks, buddy. Thanks, you too. Nice job.